by Vishnu Institute of Pharmaceutical Education and Research. I'm Sahitya, your host for today. We are privileged to have among us today's a distinguished keynote speaker who have graciously accepted our invitation to share his insights and perspectives. His wealth of knowledge and experience will undoubtedly enrich our discussions and inspire us to think critically and act decisively. Ladies and gentlemen, please join me in welcoming Dr. Surender Singh Jadhav, postdoctorate, South Korea, scientist, CSIR, Hyderabad. Over to you.
Thank you. Mm. Really, I'm comfortable. Okay. That's it. Mm. Yes. So I am audible? Yeah. But if you're not comfortable. Yeah, fine. So we can take this. You have internet? In, can you play? Go to ICT website. So good morning all. Yeah, my name is Surendra Singh Jadhav. So thank you for inviting me. Uh, Actually, I feel this institute as a, I am not a guest, but I am a like, I feel like, uh, is it audible? Yeah. So I feel this institute as a, my host actually, because I worked here for almost 1 year and I was enjoyed a lot here. Not only enjoyed, I also did lots of hard work and I also motivated lots of students and that students already passed out here last year. So thank you, sir, for inviting here. And good morning, sir, in charge principal. Good morning, madams. And I am not finding some other head of the departments here, to whom I know very well. So yeah, yeah. Can you play IS? Yeah. So I want to show small video. So where we are located in IICT. Go scroll down, scroll down, scroll down, scroll. Yeah, that video. So So yeah, thank you. With this short introduction, I thank Dr. Rajendra Prasad, Insurance Principal, right now here, and I also thank Dr. Ramesh, the principal, actually who is not here. So now I have two lectures here. So I will start with like transformations of NCs in medicinal chemistry. So I will not bore you too much. So whatever the NC, whatever I am showing here, that is the topic name is transformation of approaches in discovery of NCs in healthcare. So before whatever the drug comes into the market, that is actually coming from NCs. That is we call new chemical entities here. Okay. So how we have to identify and how we are getting NCs from the different sources and how we are designing, how we are making them using some different computational approaches. Those things we will going to discuss here. So these are the contents of my topic today. So we are discussing point by point here. Oh, here. So coming to the first topic here. So medicinal chemistry. So it is a branch of science that deals with the discovery or design of new therapeutic agents and their development into the useful medicines. So whatever the mention, I have taken this from the scientific literature only, whatever the definition of the medicinal chemistry. So most of the students might not know right now. I think they are first years or second years, but you have to be very cautious while going through the medicinal chemistry. It is very fundamental because whenever you are going through the medicinal chemistry, you should link that with the pharmacology, then only you will get idea. So both the terms, whatever the you are getting names of the drugs in the pharmacology and medicinal chemistry will be same, but pharmacology speaks about the mechanism of action and medicinal chemistry speaks about only structures and their optimizations. If you link both, then you will get fundamental idea how it is going to work into the body. So yeah, what happened? So yeah, so the medicinal chemistry it involves around there are four topics here. So one is like compound, compound or drug. So whatever the compound coming to the market or coming to the literature, we can call it as compound or chemical structure or ligand or pharmacophore. Anything we can call that is compound, and that compound will be converted into drug. And second one is structure activity relationship. How the structure of the chemical gives activity and what is the relationship between the structure and activity that can be related by the structural activity relationship SAR. So these things you will be going through, and also receptor interactions, how the drug binds into the body, different sort of proteins or mechanism of action where it goes and exerts its site of action, and also admit properties like pharmacokinetics. So this is a compound. This is the abbreviation I have taken from the pharmacopoeia. Here, a drug is a substance that, when inhaled, injected, smoked, consumed, absorbed via patch on the skin or dissolved under the tongue, causes physiological change in the body. So all routes I hospital or anything. So doctors generally will give the drug in these routes only. Other than not these routes, it is not there. Okay. So before that, you must know the sources of drugs. So sources of drugs are like natural, animal, mineral, synthetic, semi-synthetic, microbial sources, genetically engineered. So natural means most of the anti-cancer drugs are obtained from the natural. So this is classical example. You will go into the your pharmacology means medicinal chemistry textbooks. You can find all these classical examples. For example, animals means all these are enzymes. Like for example, here we have mentioned like different fibrinogen factors, antithrombins, and cholinesterols, and minerals. Minerals also like whatever the different type of, for example, rock salt. What is the use of rock salt? You know. So like that, minerals will be there. And coming to the synthetic, semi-synthetic means synthetic. We will isolate some compounds from the natural sources, and after that we will subject it to chemical modification, and that chemical modification will be semi-synthetic analogs we can call it. For example, opium, morphine, codeine, all these were isolated from the natural sources and they were modified chemically, and those chemically modified forms of things are very like highly pharmacologically active compounds. And for example, here microbial sources. So penicillins are examples. And genetically engineered means we have already know, we have passed through in the COVID gave good example. And whatever the vaccines were came, they were genetically engineered here. So coming to the structure activity relationship we mentioned, the second here. So any given chemical structure has three different properties. One is molecular interaction potentials, stereochemistry, reactivity. For example, molecular interaction potentials means you must remember the compound, whatever the chemical structure is there, whether that compound has hydrogen bond donor capacity or hydrogen bond acceptor or charge transfer, dipole moment, hydrophobic potentials, negative charge, positive charge. For example, here, these are the color indications you can see. This red color indicates that it is negatively charged. This blue color indicates positively charged. and whatever the like gray color indicates is hydrophobic potential. These kind of things you have to memorize whenever you are see the structures here. And these are all the interactions how we are mentioning here. So like hydrogen bond donor acceptor and charge transfer, like dipole moment and hydrophobic potentials, negatively charged and also positively charged. By this way, we can see all these things using the computational tools, whatever we have. And also stereochemistry. Stereochemistry, you must remember what is the hybridization state of the chemical or drug or conformation, whether that is belongs to RR or SS or configuration. And coming to the reactivity. So reactivity also very important while determining the potency of the drug here. Tautomerization, means reactivity is tautomerism, functional groups or pKa values. So we may know some of the drugs has ketoenol tautomerism, and due to that impact, that solubility profile of the total drug will be changed. It means sometimes it will be absorbed in the body, sometimes it may not be absorbed. So those kind of things also we must remember while designing the drugs. And also functional group modification. For example, here enol form, keto form, lactam form, lactone form, or lactin. And also pKa also plays very important role. Here 2-hydroxypyridine, 3-hydroxypyridine, 4-hydroxypyridine I mentioned the whether all belongs to the same hydroxypyridines, but their pKa value drastically changed. You can see here, and this pKa is directly proportional to the solubility of the drug. So whatever, that's why you always remember how pKa affects the like solubility of the drug. Wherever if you are going to the industries, for example, if you are going to industry, you have got one drug and you want to develop the method or you want to formulate the drug and you want to identify the drug into the through using HPLC. And which type of solvent you have to use, you don't know. Which type of pH you want to use, you don't know. In that case, if you know the pKa of the compound, and that pKa will help you to identify the solubility and drug prediction property using the HPLC. These things you must remember. So coming to the receptor interactions here. So that's why whenever this whatever the compound goes into the body and it reaches to the system, it reaches to the site of action. Site of action will be enzyme. Site of action will be enzyme, protein, receptor. Anything. All are made up of amino acid residues. And those amino acid residues have like positive charge, negative amine group, carboxylic group through different backbones, and those backbones or side chains are responsible for interacting with the whatever you are taking drug which went to the site of action, that is receptor or protein. And those protein sort of reaction interactions you must remember. For example, if your compound has like pi system, and which amino acid residue will interact with the pi system, you must remember. There are only 21 residues, amino acid residues in the body. 21 or 23? 21? 23? 21. In the 21 amino acid residues, which residues contain aromatic group? That's why you have to remember like tyrosine, tryptophan, phenylalanine. There are only 3 residues and as well as histidine. There are only 4 amino acids which presents pi systems. And those pi systems whether will be interacting with the drug or through pi-pi interactions, you must remember. In the same way, you can also see the hydrogen bond capacity. So which for example lysine or arginine or some sort of like aspargines, those contains amines, free amines, and those free amines will be responsible for interacting with the whatever drugs. That's why you must remember cation interaction, pi cation system, hydrogen bonds, pi interactions, and all this will be interaction between the like residue and drug component. So that's why whenever you are designing or whenever you are going through medicinal chemistry, for example dopamine or acetylcholine or any other thing, acetylcholine we are calling. So what is the specialty of acetylcholine? You know, that is a quaternary ammonium salt. Due to that quaternary ammonium salt, it will interact. That quaternary system will create a positive charge, and that positive charge is very essential to interact in the like acetylcholine receptors. So that kind of strategy you have to develop while reading the pharmacology and medicinal chemistry. Otherwise, it's impossible to memorize the drug's nature. In the same way, yeah, this whatever the secondary structure loop helix I will discuss in the coming slides. So coming to the new chemical entity, earlier we have discussed about drugs. So what is the new chemical entity? So this is the definition, proper definition given by the FDA. So what is FDA? So whether that is exist in India, US. So we have to follow US rules and regulations. Why? Because we are generic makers. We are generic makers and we are number one in the generic India, and we were focusing our generics to export to the US countries. That's why we have to follow their guidelines. And according to their guidelines, what is the definition of the FDA means here? It is a new chemical entity, that is a drug that contains no active moiety that has been approved by the FDA in any other application submitted under Section 505 of the Federal Food and Drug Cosmetic Act. So this is a proper definition of the new chemical entity. So all people will talk about NCE, NCE, NCE. Means what is NCE? That you must remember. And what is the definition? And in which country you want to file a patent. So according to that country needs, we have to focus and we have to generate. So new chemical entity NCE means don't see the question face mark. So that is chemical structure only. New chemical entity means that is a chemical structure. Okay, because that is identified purpose, different purposes like treating disease or treating infections. Okay, and these are the new chemical entities or compound emerge from initial phase of the drug discovery. So they could be new chemical structures, they could be interacting with different biological target. That different biological target will be enzymes, proteins, receptors, anything. Okay, and also they have better therapeutic responses. And NCEs after clinical trials could be converted into drugs, and they can be developed from the natural sources and synthesis in the laboratory, like sources of drugs. The same thing here. And coming to the like new chemical entity here, the same sources are, they are plant sources, microbes, animals, and also synthetic compounds. We can go for modification, we can go for combinatorial synthesis, and we can design through drug design topics here. And all these things I will explain you in very short. Okay, so before that thing, so what is hit, what is lead, what is preclinical candidate, you must remember. Okay, so what is hit? It is not a rat killer or it is not a mosquito, what that called, that is not a hit. So here in drug discovery, what is hit, you know? So whenever you don't know, one disease came, for example dengue, for example chikungunya, it came into the like epidemics, you know, epidemics, endemic, pandemic. So what is the difference between epidemic and endemic and pandemic? That's why you must remember these the terms. Epidemic means it is onset occurred at that time or during frequency time of thing, and during that time it went away again, after some times again it came. That is epidemic. It is like a guest for some time. It is coming and going. And endemic means it is a permanent guest and it came to all places. And pandemic means it is across the globe. That's why COVID time pandemic color tone. Pandemic tone means that is the same. That's why you must remember. And those side of diseases when comes into the society, during that time you don't know how to identify the compounds or how to identify the drugs, and whatever the like chemical you have identified for normal purpose, using some different approaches will become hit. It means small chemical quantity which is acting on the drug with very minimal modification, and that will be converted into lead by adding different functional groups to the whatever the chemical structures you have identified, and after that it will go under through the animal studies and human trials and it will become preclinical candidate. So these things we will go. This is the traditional drug discovery here. So in traditional it means in olden days. In olden days means 100 years back. So how they were using? They were using plants for treating the diseases. So plant constituents, we don't know which plant. Only plants they are using. So after that they were identified active chemical constituents from the plant parts, whether that is leaf, root, or stem, or bark, anything. For example, cinnamon. They are isolated. What is the cinnamon active constituent? Huh? Cinnamic acid, cinnamaldehyde. Okay, quinoline aldehyde. So quinoline alkaloids, they were isolated. Okay. So coming to the after that isolation the active chemical pointer, they were started in synthesis in the laboratory, and there after that they are optimized. So this is the traditional drug discovery process here. It means it was with knowledge base only, because they were using plants for treating diseases, and from that plant they know to treat which disease by using which plant. Accordingly, so in the modern drug discovery process, first we have to identify the target. If you identify the disease, in that disease which target you want to focus to design the drug. So after that you have to validate the target, where that is validate means the target is correct, accurate or not. For example, gene knockout studies will tell you the target validation. For example, if you are targeting gene A that is causing cancer, and if you knock out that gene in the mice, so that gene will be gone. So if you target that gene, whatever identified will not work. So by that way, they will validate the target here. And coming to the like hit discovery phase, once the target identified, validated, then new chemical entities will be started for discovering here. And hit to lead phase, this is the all this can be done by this is RDD, rational drug design. It means knowledge based design. So knowledge base, how you will utilize your knowledge to design the drug. After the structure base, ligand base, fragment based, and as well as AIML. So all these things will be discussed here in brief today. So this is, I am not going to extend too much. This is how we have to design the new chemical entities. And coming to the general CAD approaches here. So this is the traditional drug discovery process. Like that, I have shown traditional drug discovery process. From that traditional drug discovery process, the target identification approach has been discovered. So the target identification, once you identify target, you can go to design the new chemical entities by using two different approaches, that is structure based and ligand based. And this structure based means here I am talking about the structure of protein. Protein means the target may be disease protein or receptor or enzyme. So what is the difference between protein, receptor, enzyme? Proteins has different role, receptors has different role, and enzymes has different role. Ion channels comes under receptor or protein? Receptors. GPCR, G protein coupled receptors. Receptor. What is the role of the G protein coupled receptors? Signal transduction. If something happened here, it is going signal to the brain. So that kind of signal transduction will be occurred through receptors. And enzymes, for example, lipase, hydrolase, pancrease, all enzymes are responsible for enzymatic activity, means decomposition of metabolites, and also ion channels, transporting of ions from one place to another place. So all this can be done by structure, means all those are made up of amino acid residues, receptors, whether ion channels, whether enzymes, all made up of amino acid residues. Once you know the protein structure whole, you can go and you can identify the protein, you can identify the pocket of the protein where you can design the drug and how it binds, and how the protein structure can be determined. It can be determined by X-ray crystallography. So X-ray crystallography, if you take some protein in experiment in the some lab wares, and you will go to the instrument, and that X-ray crystallography will tell you the like body X-ray how you will done the same fractures. The same way protein fractures how it is there, it will show total real crystal structure of the protein. And NMR spectroscopy also can tell the protein structure, and also cryo-electron microscopy. So this is the recent technique got Nobel Prize in this area. Okay, and also ligand based. Here the knowledge of chemical structure. You have to remember this is the knowledge of protein and this is the knowledge of chemical structure. So you can design the drugs or NCEs by two ways, by focusing on the protein or by knowledge of the chemical structure. That can be like hydrogen bond donor acceptor capability, lipophilicity, size of the molecule, charge, and electrostatic forces. How we can measure the size of molecules? How? That's why it is very important. Angstroms, you know angstroms units. So we have to measure the size of the molecules or the size of the proteins by angstroms. So these are the fundamental questions, and these structure-based techniques can be done by these techniques here: ligand docking, homology modeling, fragment-based design, or molecular dynamics. And ligand-based techniques can be done by pharmacophore mapping or similar QSAR and similarity search. Okay, so these are the structures of the drug which are identified by some different approaches. Structure-based drug design has yielded this darzolamide drug in the market in 1995, and here Novartis has provided imatinib mesylate, which has optimized through rational design, and a melanoma cancer drug here it has optimized through fragment based design. So each and every technique has their unique specialty to afford the drugs into which are came into the market. So coming to the all these things, you must remember whenever you want to do all this sort of work, you have to get the knowledge of chemistry, you have to get the knowledge of specific target or specific compound, and also you have to identify the biological active target, and also compounds you have to identify, and also knowledge of ligand protein interaction, how it interacts with the drug and receptor. And also you should have the intelligence of medicinal chemist, how you will intelligently sort out that problem. And also you should have computer knowledge, some little bit computer knowledge, then only you can carry out all these things. So coming to the example, say mechanism based here, NCE, I am talking about NCE, it means new chemical entity. Here I have provided you the HIV protease. So HIV protease, it means that protease enzyme is responsible for processing of viral protein into the different other fragment proteins. And how it is, so whatever the drugs here you have mentioned here, ritonavir, saquinavir, if you go into the market, you can identify ritonavir and saquinavir are using to treat the HIV. HIV is also called as AIDS. Okay, and also how they are discovered, you must remember, you have to get your question, you have to question on yourself how they were discovered, because they were discovered through mechanism. In HIV protease, this is the sequence, you can see substrate here. So in HIV protease, aspartic acid residue are present in the HIV proteases. These two aspartic acids will sense the signal of the phenylalanine. People have identified that these two residues are very important amino acid residues, and if you focus on the residues, and they were created additional hydroxyl group here. So they were created additional hydroxyl group in between these two residues, and those hydroxyl groups you can see here everywhere. And those due to presence of those hydroxyl groups, that replication process of the protease activity of the HIV was stopped, and these drugs become fundamental in the treating of HIV positive patients. So these sort of things you have to memorize while going through the structures of the drugs. So coming to the HIV, the same thing we can here call it as transition state mimics. For example, these all are marketed drugs. I mentioned here saquinavir, nelfinavir, fostempiravir, darunavir, indinavir, emprinavir. So all these are veers. You can see veer. All these veers are useful for treating the HIV patients. In all these drugs, this is the unique drug. You can see atazanavir. This is unique behind all these things. Why? Because it is having hydrogen moiety here. You can see acyl hydrogen. This is hydrogen, NH2. NH2 is hydrogen, and behind that position acyl group is there. That's why we can call it as acyl hydrogen. So if you see, all other compounds doesn't have acyl hydrogen, but it is having acyl hydrogen. And you can see here their overlap in the body when it goes into the receptor site of action, and you can see their overlap. This is indinavir. This drug is indinavir, and they both overlapped, and their 3D complementary shape you can see here. This is NH, this is another NH, and they were mapping with the some sort of mechanism. So by this way they were identified the drugs which are treating for the HIV, because HIV keep on mutates, and that's why we cannot completely eliminate that disease, but we can identify new drugs to overcome that mutations. And by pointing that mutation terminology, people are continuously discovering the new chemical entities, and those were becoming the drugs. And this is the another target of the HIV here I'm talking about, INST. So what is INST means here? Integrase strand transfer inhibitors we can call. Integrase means, for example, HIV is RNA or DNA virus. That you have to memorize. That is the RNA virus. Okay. So HIV's genetic material will be integrated with the human DNA. So that integration part will be taken care by the HIV integrase enzyme. So that HIV integrase enzyme, what it does, it integrates the HIV genome with human DNA, and that's why if we stop that integration process, we can completely eradicate the HIV infections, or we can drastically reduce the HIV count. And that's why here you can see this is the diketo acids here. This diketo acid, this is a fundamental molecule. Some people were saying cinnamic acid. In the, if you go and if you add another hydroxyl group in the cinnamic acid, it can become diketo acid. And these diketo acids are responsible for interacting with the metal ions present in the HIV integrase here, one magnesium, another magnesium. And this sort of system will it will form covalent bond in the integrase, and based on that mechanism people were identified first generation drugs, that is raltegravir, elvitegravir, second generation drugs, and also now it is in next generation phase. Some people, some of them are identifying, try to identifying new sort of drugs. Why? Because this class of drugs are very important. It means the newly identified HIV positive individuals, newly HIV positive individuals. Why? Because I am saying newly infected persons will be given treatment with this sort of drugs only, because initial phase of HIV infections can drastically can be reduced by this sort of drugs. So that is very important here. And these are the mechanism how it is binding. Here you can see these diketo acids here, they how they will bind with the metal ions. Here you can see metal ions, so they will form the covalent bond, so non-breakable bond. If it forms non-breakable bond, so it may not integrate with the HIV like human integration here. So this is some other sort of example. These are some different example. You can see oxycam, diclofenac. So what is the specialty of oxycam and diclofenac? People will get body pains, go medical shop, cam. But where they will bind into the body? You know, COX, cyclooxygenase. So cyclooxygenase has two different isoforms, COX1 and COX2. And these whatever the drugs which are there in the market, they were non-specific. Whether they are going to bind into the COX1 or COX2 were not identified properly. That's why they were drastically giving side effects. What is the side effects of the COX inhibitors? Don't know. Gastric irritation. Gastric irritation is the side effect. And why that gastric irritation giving the side effect? Because that may be acting on COX1, and the COX1 is highly present in the gastric mucosa, and that's why irritation is occurring. So if you specifically identify the COX2 inhibitors, in that case you can drastically reduce the COX1 side effect. So by this way, some people can also focus on the like oxycam binds in the COX pocket like this, and diclofenac binds in the pocket like this. And if you can identify the total combination of these two, so we can identify how it totally occupies in the pocket. So L shape. So this kind of strategy you have to develop by using your knowledge. So coming to the this slide, I will explain later. This is for example, this is chikungunya. You know chikungunya? I think nowadays people might not know, but like 10-15 years back, people were infected with chikungunya virus, and side effect of the thing is swollen joint pains, drastical, very like unbearable joint pain, and that unbearable joint pain is happened due to virus. And how chikungunya virus spreads into the like humans, you know? Don't know. That's why you have to get knowledge. That is the cause of chikungunya virus to the human is mosquito. Simple mosquito bites, that virus comes into the body, and it will create lots of problem to the body. It means it will go and it will sit on the like joint pains, and it create lots of joint pains. And this is a protease. So this protease enzyme, it is there till now. Chikungunya is not having treatment. Treatment ledu. Okay, no treatment is there, but we know how the viral protease is responsible for chikungunya viral replication. If you know the viral protease role, then you can target and you can stop the replication process, but that is huge. It will take lots of time. So by using that thing, we can screen the total databases. What is databases? Means the physical compounds, you know, chemical structures in your laboratory, lots of chemicals will be there, but all are physical compounds. It means you can touch, you can feel them. But databases, so databases means they will be digital compounds. It means lakhs and millions of compounds will be databased, and the structure we can directly use to screen the protein, virus protein into the using computers. Computer drug design means screening of the target with different sort of, is we are screening those databases targeting the target of whatever interest to identify the different compounds, chemical structures. Once if you identify the target and if you identify the compounds, and you can directly test them, you can buy or you can synthesize them in the laboratory, and you can directly test them biologically, and you could identify the like their mechanism of action. So this is how, and this is also case example dengue here. So you know dengue story? Don't know? So dengue, how many serotypes are there? We are pharmacists here. We have to be always compete with literature. The society, not Facebook, not Instagram, not anything. We have to always focus with the scientific literature. So dengue has 4 different serotypes. Among them, 4 serotypes. Serotype 2 is very dangerous, which is very prevalence in the India. So whatever here compound we are mentioned here, all are responsible for dengue viral entry inhibition. So dengue is also spread through mosquito. Okay, that mosquito is causing again dengue to human beings. That dengue will be landed that virus into the humans, and that human again that virus requires it to enter into the host cell, and that host cell and that viral integration will be can be done by glycoproteins. We can call in COVID spike protein. We call here glycoproteins, and that glycoprotein is responsible for viral entry into the host cell. If you block that glycoprotein, then viral entry into the host cell will be blocked. And all these compounds were identified through different computational approaches, and also they were synthesized and biologically tested, and they were successfully become dengue viral entry inhibitors. Viral entry inhibitors I am talking. Why? Because they are resisting the viral entry into the human system. So coming to the ligand based here, for example, ligand based means this is a QSAR example, quantitative structure activity relationship. All these things you will come into the second year or third year of your syllabus in medicinal chemistry. Okay, that's why it is very important. So quantitative. So what is the difference between quantitative and qualitative? Don't know. Qualitative means approximate. Quantitative means accurate. Quantitative means accurate. Qualitative means approximate. It means that approximate should word will not cannot use in the quantitative structural activity relationship. It means which functional group of the chemical compound is accurately inhibiting or responsible for pharmacological action. That we have to identify accurately. Then only we can design new sort of compounds. For example, here these are all kinase inhibitors. You know what is the role of kinase? You don't know kinase? Don't know? You know ATP? ADP? What is ATP? Where it binds into the human? What is the why ATP is very important? That energy will be transferred by kinases. You know anti-cancer drugs? What are the classification of anti-cancer drugs? That is different. Now go to literature and go to search WHO, World Health Organization classification of anti-cancer drugs. There you will identify among 100 anti-cancer drugs. Among 100 anti-cancer drugs, there are 80 kinase inhibitors approved in the market. Belongs to kinase inhibitors only. More than 80. More than 80 drugs are belongs to kinase inhibitors only. Why? Because all kinases in the human, there are more than 400 kinases were discovered as cancer causing. There are more than 500 kinases are discovered for cancer causing, and those 500 kinases are different ATP. In every kinase ATP binds, and that's why if you target the ATP, adenosine, you are talking about adenosine. What is the chemical structure? You must remember. If you know the adenosine structure, you can mimic that. This is a sort of pyrimidine here. You can see pyrimidine. This is, you can see pyrimidine. You can see pyrimidine, and this pyrimidine core will mimic the adenine core of the ATP, and it will competitively bind with the ATP pocket in the kinase. And once it binds into the ATP competitively, then energy transfer of the cancer cell will be blocked, and that's why anti-cancer activity will be exerted. And this all designed through some different sort of kinase inhibitors. If you know the knowledge of kinase inhibitors which are present in the market, if you do the functional modifications in quantitative manner, you can identify easily new chemical entities for treating the cancer. Similar way, what is the, the sildenafil citrate is popularly known as Viagra pill. Okay, popularly known as Viagra pill, which were identified through QSAR techniques only. Okay, and it became black box drug. So it is a PDE5 inhibitor. So PDE5 inhibitor, you can go and search in the literature what is PDE4, PDE5. So that's why whenever we are talking about new chemical entities drugs, we have to definitely correlate its structure and also pharmacology. Then only you will get at least interest. Otherwise, this will be very difficult. Okay. So the same way, these two are PDE5 inhibitors, popularly useful for some reproduction activities. So coming to the ligand based here, bioisosteer. You know isosteer means what? Isosteer. Don't know? Bioisosteer. That's why isosteer means chemically relevant. Chemically relevant or functionally relevant. For example, you can see here, this is the benzthiazole. This is also benzthiazole. But you can see here, this is triazole, 1,2,3 triazole. It can be converted to into 1,2,4 oxidazole or 1,2,4 oxidazole means equivalent. Chemically, they were equivalent, and also if they are chemically equivalent, it means they also can exert similar sort of interaction in the receptor with amino acid residues. That's why isosteer we call bioisosteer. It means biologically they will exert similar sort of interaction with the receptor, but their chemical structures are different. Their complementary shape will be same, but chemical structures will be different, and they will exert similar sort of action with the amino acid residues of the proteins or site of action. That's why if you know the different sort of bioisosteers, you can modify them and you can simply create new chemical entities for treating same disease. For example, I have shown in you the HIV protease inhibitors. So coming to the another topic here, artificial intelligence in drug discovery. So here this is very important tool here in the current era. So artificial intelligence is the simulation of the human intelligence processes by machines, especially computer systems. Often AI is simply component of the technology such as machine learning. So what is machine learning? Machine learning people will talk. So to do machine learning, you have to remember you must know Python script. Otherwise, you must be master in the R language or Java or C++ or Julia. Then only you can use AI by ML techniques. Otherwise, it is very difficult. Okay, as pharmacists, we are pharmacists, we should not be master in all aspects. But you have a good chance to create yourself where you want to stand. That thing you have to question your mind, and accordingly you have to choose your path. So it requires Python coding, R language, Java interface, C++ basics, and Julia. So here artificial intelligence development chart in 1950s, it was discovered by Alan Turing, and right now we are employing in different sort of applications in almost all applications, including cell phone to diagnosis. Everything we are applying using artificial intelligence. You are now seeing or watching your mobile phones, and all that is also uses artificial intelligence tools. Why? Because that much popularity it gained. Why? Because simply it was because databases, only databases. Whatever the earlier textbooks we were having, those textbooks and those sentences were digitalized, and it was digitalized, and those digitalized words were trained by the computers. And how computers trained by because they were trained by different deep learning approaches, machine learning approaches, and reinforcement approaches, and all these approaches are comes under artificial intelligence. Once the computer is trained in every sequence and every sentence and every phrase of the human beings, and it become very easy, and we become very lazy, because computer becomes smart and we become very lazy. Why? Because it whatever the things we want to do, it is doing by computer only, including driving of the vehicle, self-driving cars we got tracking everything. So those type of development chart also we can use in the drug discovery processes here. Okay. So coming to the artificial intelligence, it means these are the three different learnings and three different phases here. Deep learning means it is a subset of machine learning, and machine learning means this is a subset of different algorithms, and AI means it is a combination of ML and deep learning. So this thing I am not going to take too much time of this thing because you may not understand, but this is a hot topic. Wherever if you find time, you can spend some time how to do AI ML tools or how to initiate your studies, because you are B from like final years or second years or third years, but right now it is a good chance to you to initiate these things. Why? Because you will get lots of chances. For example, if you want to do masters in different IITs, NITs, or NICERs, or ICERs, or NIPERs everywhere, they will facilitate you to learn on your own. If you want to learn on your own, you must remember what is the literature going on with the scientific society. You have to get update every day. Otherwise, it is very difficult. Okay. So coming to the like application of AI, it can be applied in the e-commerce. E-commerce means Amazon, Flipkart, Meesho, and popularly I don't know all shows are there. And also like smart cars, healthcare, virtual assistant means Siri or Google. I don't know Google what I call. And also hospitality. It means if you go in some everywhere tourism or anywhere, if you sit in the bus stations everywhere, you can see the time charts or you can see the schedules. All were totally done by AI tools. And also surveillance, security, customer support. If you want to reach customer support, you have to wait for 5 to 10 minutes, and that 5 to 10 minutes waiting period is done by AI tools only. Okay. And also social media marketing, everything. And but in health aspects, you can see here how AML is very important. This it is useful for target identification. It means most of the innovator. What is the innovator company and generic company? You know, innovator and generic. Don't know. Innovator means the innovator companies generally develop the drugs. Innovator companies will identify new drugs for diseases. They will develop from the drug discovery phase. It will take 15 years to reach into the market, and last 5 years they will market. And during that last five years time of point, other companies may not have rights to sell that kind of drug. And those innovator companies, how they will identify the target? They are using the AI tools, and all Indian companies are generics. It means we are making duplicate drugs. Though they are same sort of, they are giving same sort of mechanism of action, but our drugs are very cheap because we are not doing development. We are doing generics. We are copying. We are not doing. Okay. So this is a target identification or molecular generation. So we can use in the preclinical research and also clinical trials, drug efficacy, adverse events, and also biomarker detection. So drug efficacy means you must remember how much quantity of the drug is required for human dosage, like 500 mg, 650 mg, 300 mg. You know the dosage, that is called efficacy. That much dose is steady for you or not. So that kind of efficacy study also we can do by AI. Adverse events, what is the toxic parameters causing by the drug, also can be determined by AI. Biomarker detection, what is biomarker? What is biomarker? Don't know. Best example, if you are coming to the classes, the person who is coming every day late, he will be called as late Latif Khan. And biomarker, now they have given him biomarker because he is always late. In the same way, in the gene level also, one gene is dysregulated, people will identify that gene and they will put a biomarker on that gene that is causing some trouble. So that kind of thing can also be done by AI. And also here, patient care, it means drug prioritization, patient intake, patient readmission. Drug prioritization means, you know, now system how it is changed, it means people will get cancer. Okay, now we have different continent people's different continent cancer patients are there. There is a cancer consortium. So now that cancer consortium, what it has done means, it is getting the total patient samples and it created database. And with respect to demography, with respect to geography, with respect to male female ratio, and with respect to cancer type of cancer, it created database. And that database now is giving the doctor information that which kind of drug is suitable for which kind of patient. One drug cannot be given to all cancer patients. Some patients lifestyle will be different, some patients geography will be different. According to that sort of parameters, drug prioritization also is coming into the picture right now. Okay, coming to the diagnostics, dermatology, radiology, pathology. So all this can be done by AI tools right now. So these are the advantages and drawbacks. It can reduce the human error, it can quickly make the decision, it can reduce the risk. And these are the negative things with the AI. It is costly, it causes job loss. If everything is doing by computer, then what we are doing? That is a question mark here. And also, it is also reducing the human ability, thinking ability. It is, for example, us and now next generation. You can see their intelligence that can be easily correlated by the right now comparison, okay, and also emotional, it doesn't have emotion. AI tools will doesn't have emotion. If you call customer care repeatedly, sorry, 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 it will say, but it will never say I'm happy, I'm happy, I'm happy. Okay, the same way it will also enable it to integrate the ethical principles. It doesn't have ethical principles. For example, chat gpt chat gpt, if you are working on kindness, she is also working on kindness. You ask kindness, you talk about kindness, it will give you one paragraph, it will give you another paragraph. It doesn't have ethics, but it will not tell that I have given to him or her. It is giving its job, that's it. It means this kind of things are there. AI in drug discovery, it means we can monitor the reaction outcomes, nanotechnology, wastewater treatment, molecular design, drug discovery, retrosynthesis, natural language processing, molecular properties. All these things are there, and also machine learning here. Machine learning and same thing I am talking. So this is the industrial parameter. I will carefully example you. So finding materials and consumables. You want to synthesize paracetamol. For example, in industry, you want to synthesize paracetamol, and you have hundreds of chemicals in the warehouse. Where the starting materials of the paracetamol is present inside the warehouse, you don't know. You have to manually identify, otherwise you have to systematically maintain the repository. In that case, if you manually identify and if you make the database, it will identify the materials and consumables required for paracetamol synthesis, and it will tell you where you go to the location, you go this much grams of starting material, and you react in this environment, you will get paracetamol. This kind of things will be done by AI. And also chemical synthesis, the same way, data mining and oncology. So data mining means, for example, different cancer patients have different mutations. For best example, colon cancer. Colon cancer, you know what is the specialty? The largest organ in the body is colon, and colon cancer is also there. It will present in the upside of the colon, and it will go into the wherever it wants. It will travel. Okay. So, for example, in that cases, which part of colon cancers, which type of colon cancer, which type of mutation. So that kind of, if you have the information, you can easily mine that information and you can create the drug discovery process here. And also biological validation here. Biological means target. If you identified the target of disease, you can validate that thing. Lead discovery, it means chemical identification. Biomarker, that's why I mentioned cancer causing or some disease causing genes. Clinical development of the drugs. Pharmacovigilance, it means it is also sort of databases, means post like post clinical trial data monitoring type of thing. Drug repurposing, you know what is the drug repurposing means? You don't know? Yes or no? What is drug repurposing? Getting more all sleeping or all busy with smartphones? No? What is drug repurposing? Clinical trials. Then already established drugs for other. The same way. So drug in COVID time, people were in a huge demand that they want to identify existing drugs to cure. For example, best example, how many drugs present in the market? You are a pharmacist. You must remember, you should not count. You should say approximate or accurate. That's why more than 2,500 drugs are present in the market. More than 2,500 drugs present in the market. In COVID time, people were purchased all drugs, physical drugs. I'm talking, not databases, physical drugs, and they were screened the virus in all aspects. And all people are telling that this drug is working, this drug is working, this drug is working. Why? Because drug repurposing, the advantage of drug repurposing is it may not require clinical trials. It will save the patients, it will save the drug discovery time. It means it will save your 15 years time. It will not cost that much huge amount of the money to the company to identify the same drug for different purposes. Okay. And also virtual lab assistant, the same thing, this is computational thing. All this will be coming again to clinical trials here. So these are the best examples. This is the picture. I have not done everything, but I am showing the glimpses of the picture. So this is the Nature Communications paper, network based prediction of drug combinations. It means we are some most of the diabetic patients are there. So they will use different combination drugs, metformin, fenformin, something. How that drug combination works? Yeah, tea break. Just I will take you 5 minutes. Okay, is it fine? 5 minutes? Yeah. Okay, you can see the drug combinations here, and also some different sort of disorders, and also some diseases, drug interactions, and efficacy screening. All this can be, you can see, and you can go through the literature, then only you can identify. The ultimate task is you have to compete with literature, then only you will get idea. You should not compete with newspapers. Today is holiday, tomorrow is holiday, day after tomorrow is holy. You should not compete that thing. Okay. So here, before going to the like protein drug protein interactions means amino acid residues. In general, proteins, secondary structure, primary structure, tertiary structure we call. Primary structure is the protein sequence, means amino acid sequence. It is starts with some different. Okay. Secondary structures means helix, loops, and sheets. And tertiary structure means it is a combination of multiple secondary structures. And also it has, if you know the quaternary structure, then you could identify the protein active pocket. So this way you can see and you can go to the literature. This find site super combination is there. It is doing all sort of activity. It is free of cost here. So all these things are there. You must remember. That's why protein is knowledge is very important. So you should be a dynamic in this knowledge. So and also this is the literature I am mentioning here. So AI calculated biomarker cancer. This is also I have adapted from the this sort of literature here. How they are doing you can see. And also this is the same thing I mentioned here. So I am not going to go through this thing. And this is also some different sort of machine learning algorithms. This also you will not understand. And also you see here machine learning methods in drug discovery. You can see protein-protein interaction. Why protein-protein interaction is very important means one protein interaction with another protein. For example, I was talking about colon cancer. So colon cancer spreading, it means spreading of colon cancer from one part to another part takes energy giving source to the cancer cell. And that energy giving source means it is interacting one protein with another protein. Then only it is getting interaction in energy. That's why it is transferring from one place to another place of the colon. If you stop that protein-protein interaction, you can stop the colon cancer spreading. That's why protein-protein interaction is very important. Those can be learned by machine learning. And de novo design. You know what is the meaning of de novo design? Alar Telugu? No Hindi? ప్రపంచంలో లేని కొత్త కెమికల్, లిటరేచర్లో లేని కొత్త కెమికల్. It means the chemical which is not exist in the literature, it is not exist as a physical form or any databases. That kind of chemicals you have to design, you have to synthesize, and those kind of de novo design can be done by AI methods. So these are the some different examples of the databases, natural product databases, skin sensitization database, which is whether that is itching, causing skin damaging, those kind of things also we can see using this database. This is the resource. Site of metabolism, you can see here, site of metabolism. You know site of metabolism? Where? Don't know? Liver here. Liver has cytochrome enzymes, and that cytochrome is contain, it will metabolize into two different phases, phase 1, phase 2, and that database will give you whether the drug is going to phase 1 or phase 2. So that kind of things also can be predicted by AI tools here, and all these things are there. These are some different literatures, and this is AI in chemistry. I don't want to discuss because this is how AI will understand the chemical structure. It will multiply the chemical into the different vectors, and it will give you the new chemical here. It will understand. It will give you new chemical entity which is not reported in the market but biologically similar. And these are some database. I was talking about database. So this is a database, zinc database, e-molecules database, some other databases, pubchem, and all these databases will provide you free of cost drug structures or chemical structures in million numbers if you want. Just you want to get the protein structure, you tell computer to screen these compounds and give me one compound to check the biological activity. That can be done by this sort of databases here. And this is also the framework of the AI tool. This is how AI works. If you keep on, for example, improvement, you know, exam improvement, I think intermediate, all people will write improvement. Why improvement? Getting more marks. Okay, same or backlogs, getting pass. In the same way, if you tell in phase 1 IA, it will not listen properly. If you keep on telling that, it will listen properly and it will give you accurate chemical structures can be synthesizable or testable. So this is some cost estimation. So if you want to synthesize this compound, it will calculate the raw material cost, how much cost, and it will give you the economic route of the uh, synthetic process. You can, all this can be done by AI. So coming to the, it can use computational tool, organic. Also, this is medicinal chemistry. It is admit. It means pharmaco like absorption, distribution, metabolism, excretion. All this can be done by AI tools here. So this is the best example here I mentioned. You know pulmonary fibrosis. What is fibrosis? Tissue or mass formation in the lung. It causes excessive mass. So for example, this is a drug. People were identified. This is a drug which can reduce the pulmonary fibrosis, but it is causing cytotoxic, means what that called metabolically HERC channel. Sorry, this is metabolically unstable. It means it is causing liver toxicity. And what is the difference between these two compounds? That's why you have to put brain. So people were identified that this is a site of metabolism here. This is a site of metabolism. That's why it is interacting with the liver enzyme. It is causing liver toxicity. And people were simply kept here methyl. And liver toxicity gone. And now this is in clinical trials. This is how pharmacokinetically we have to optimize the new chemical entities. We have to ensure the liver toxicity also. So we this is our lab activities, how we are going to do work in the, we target synthesis like antiviral kinase inhibitors. We also work on protein interactions and mutagenesis. We also work on agrochemicals. Agrochemical means whatever generally we are cultivating here, our societal needs, rice, tomatoes, potatoes, or red gram or any grams. So we will isolate those organisms and we will identify the new chemical entities. We will test. These are our students, and these are our mole bank compound here. Whatever these are, he is our, actually this person also witnessed in this auditorium in 2020, 2019 when I was faculty here. This person came here in this auditorium. He delivered lecture. Okay, the same way. So this is the example I was talking about. This is the colon cancer protein. When it interacts with the energy giving protein, that colon cancer migration from one place to another place occurs. If you stop this interaction between these two protein, colon cancer migration will be stopped and colon treatment, cancer treatment can be done. And all these compounds were identified by giving this approach only. Simple, they have taken the fragment of this energy giving peptide and they synthesized and they given to this protein. And once this combination of these peptides and this protein is very friendly, and due to their friendly nature, this whatever the original protein is there, that is not able to bind into the with this protein, and that friendship will be strong, and that's why this interaction will be blocked and anti-cancer activity will be gained. So this is the start. So this is the same mechanism. So these are the peptides, amino acids, but now we are planning to identify the new chemical entities, heterocyclic compounds. Heterocyclics will be not having amino acids, but here we are also preparing a small chemical compounds here. This is the kinase what I am talking about. The green color is the ATP. Here you can see this green color is the ATP, and whatever the kinase inhibitors we have identified, it means this adenine part is there, that is mimicking by the pyrimidine part of the our discovered compound. These are the total 468 kinases is there. Among that 468 kinases, it was inhibiting only specifically CLK5. CLK2 means CDCL kinase we call. So this is how it binds here in the receptor. Here ATP will bind in this pocket. In the same pocket, whatever we have discovered, this compound will bind, and that's why anti-cancer activity occurs. Logically, among 468 kinases, it is specifically inhibiting only one kinase. That is called specificity. Specific, we have to be very specific while designing the new chemical entities. The same way, you can see the some different optimization approaches. Here it has only one chlorine. We have two chlorines here. Two chlorines. Then what is the effect? Without chlorine also, what is the effect? Whether that is binding in the ATP pocket or not. This sort of activity we will do generally. This is also the same. CLK1, DIRK1. DIRK1 means that is also relative both. Okay, yaar. Why you are worrying? You know, अभी कोई आया आपके घर में मेहमान, आपका बाजू में आपका चाचा या काका या फूफी या कोई भी है, उसका घर में भी जाएगा नहीं जाएगा? వెళ్తారా వెళ్ళరా? They will go. In the same way, this CLK1 is related to DIRK1. That's why this compound is going into this pocket also. Why that's why it is also inhibiting the same, because their environment is same. It means you have to memorize the protein structure knowledge, then only you will determine fundamentally what I mean to say. So I think this is the last slide. So this is also like protein interactions. This is generally immunotherapy. You know immunotherapy, immune system. The cancer cell is there. That cancer cell will make friendship with the T cell. If they are both were making T cell, and what is the activity of T cell will be done? That is diminished. It means that false negative friendship will cause immune system avoiding immune system. And if you stop that protein interaction, you can get new sort of anti-cancer. That is immunotherapy we call. Okay. The same way this is agrochemicals we are preparing, and you have to work hard. So why I kept this picture means I met this person. You know who is this person? I think you may not know. He is a Nobel laureate. He made palladium coupling reaction. Palladium metal is there. Due to that palladium, CC bond formation will occur. Carbon-carbon bond formation occurs. And that due to that contribution, he got Nobel laureate. So I met him in 2012, 2013 in Taiwan. The same way, he is a virologist, famous virologist. Across the globe, his lab is world number 2, Rega Institute. I also met him, and you know, this is a person I have taken the good image of this person on with my own hands when I was having camera. He was looking for his lecture in the 2013. Actually, his two students got Nobel Prize. His two students. He has not got Nobel, but his two students got. It means how much dedicatedly he shined the students. That thing you have to memorize always. And also, I met this person in the one symposium in India, Jaipur, that is Vanasali Vidyapit. He is most popularly works on the like sigma receptor. Sigma receptor is belongs to the autoimmune disease. So if you go through the autoimmune disease, there you can understand what is the role of sigma receptor. And also, fortunately, I also met another person. I think you know this popular person. Don't know? Dalai Lama. He is also a famous Nobel laureate. So last year we got chance as a part of our CSIR system. So we met him in the Himalayan region. So I am somewhere here. Okay. So thank you. And see, this is my department where I have done my PhD. So this picture also I have taken on my last day of the PhD in 2015. So right now we are here. I have done my PhD here. This is a organization, but this is a department. I have done my master's here. And if you have any doubts and if you want to do anything in my laboratory, you can reach me here. And thank you. So I am sorry, I have taken, I think, 1 hour only I have taken. Actually expected 2 hours. No? If you want to ask any questions, you can ask. I am very free. I won't charge. Or if your organizer, I think they might have told you to anything to not ask questions. No? Why you are dull? Exams? Exams will be there, yeah. You will write, you will get passed. That's it. Tea break? Okay. Chalo. Chalo, we will break for tea. Thank you.
I request everyone to go for the tea break and come back here in 10 minutes. Thank you.